All right. Is there any other housekeeping items, Ashley or Heather, before we get started?
No, I would say the only other thing worth noting is we have a chat function. We'll try to keep that monitored during the presentation, and we will have time at the end for questions. We try to leave as much time for you guys to open that up for any questions you may have. We've collected questions currently from anything you've sent in. Um, so, I think if we'll try to breeze through the material pretty quickly, and then we'll open up for a discussion. If you have anything you want to feel free to put the questions in the chat or there should be a hand function at the bottom, depending on what your Zoom screen looks like, that you can raise your hand and we'll call on you. All right.
Okay, great. We will kick it off. Ashley, if you want to get us started.
Sure. So why don't we go ahead to the next slide? Sometimes we have new faces between each of our Q&As, which we have 2 to 3 times a year, depending on what's going on with the program. So I wanted to take just a few quick slides to introduce Odelia in case there's anyone new joining us today. So why don't we go to the next slide? So for those of you who we haven't met before, it's nice to meet you. Thank you for taking the time to join us today. Odelia is a nonprofit biotech. Our primary mission, and why don't you go ahead and jump to the next slide? Uh, as a 501(c)(3), we work to accelerate therapeutic development for rare diseases. We do this in a number of ways. Our primary pursuance of that mission is in the drugs we develop, which are gene therapies. Right now, those are for 3 different vision loss disorders. And our lead program, the one that's furthest along, is our RPGRIP1 program, which we're updating with you guys today. Why don't we go to the next slide? This is our current pipeline portfolio. As you can see, RPGRIP1 is that first blue bar. We're in late stage, what we call late stage preclinical testing. The program currently has orphan drug designation and rare pediatric designations, which were awarded and are designations recognized by the US FDA. We have funding through this community, and we'll update on fundraising efforts to date at the end of the presentation. But we also have um, funding from Foundation Fighting Blindness, which was a 2-year grant, 1.5 million, which we're very thankful for and appreciate you guys and all the fundraising you've done to date that helped us kind of leverage the progress of the program to get in front of FFB. I'll update a little bit on partnering efforts, but we'll come back to that. And just if you're ever curious about our other programs, our second program using the same AAV capsid, which we call ANC80, that's the part that helps deliver the RPGRIP1 gene into the eye. It's on the same capsid, but it's for our USH1C program, treating vision loss in the context of Usher syndrome caused by USH1C mutations. And then our third program, which we announced earlier this year, is on a different capsid, AAV5, so not ANC80, but closely related AAV5, and that is for vision loss in the context of MPHP1 genetic mutations. All right, so with that, why don't we go to the next slide and just another point of expansion just on our mission. So we have our biotech pipeline, 3 AAV gene therapies in development, but we also have a broader remit to take learnings from each of our programs to the broader rare disease community. So we work under what we call bridge solutions with a number of pretty diverse rare diseases, and that runs the gamut from some vision loss disorders through to neurodevelopmental and pediatric epilepsies, overgrowth disorders. And so through that, we've actually kind of been able to bring together under our mission the broader remit of accelerating therapeutic development for rare diseases. But this has allowed us really to have a broader perspective on our programs and bring learnings from other areas of drug development and rare disease back to, let's say, our RPGRIP1 program and vice versa. So we're hoping to grow this kind of rich ecosystem of sharing and get over time. All right, on to you, Jenny.
All right, so into the exciting stuff. I will be able to provide you guys with an update of the current happenings around the RPGRIP1 gene therapy program. So this will be an update since our last webinar. I believe it was in March of 25. So we have had some substantial updates and activities take place since then. So this year we've been able to complete the 50-liter toxicology grade production with Andelin Biosciences. We are partnering with Andelin to manufacture our gene therapy vector. That production is complete and all of the testing has been completed for this. And we have also developed a fit for purpose potency assay with our vendor Farmeron. And so those are two very big important activities that allow us to move forward into our non-clinical toxicology safety study. And that is the final safety study for an IND submission. And so uh, at this point, we are getting ready to initiate our toxicology study on Friday. So we've been in the contracting phase and finalizing the protocol with our vendor Versio on this safety study for the past 5 months. And so we are in a great place. We're going to kick off activities next week, and that dosing will take place early in 2026. And we should have information from that final safety study by mid-year. So all things are full steam ahead with the safety study. So it's very exciting news. As for next steps, we are going to continue developing tests that validate our manufacturing process. So there's a couple, there's one other assay that we're looking to develop and then possibly expand on our potency assay. And then we're also still developing tests or assays to measure our preclinical safety. And those assays will be developed in parallel to that safety study. And we will, once the safety study is finalized and complete, they'll be able to reproduce a full report of the findings on that study, which we can use for our IND package. And we may also engage the FDA and the community on alternative clinical trial paths early next year, just in the absence of funding. So we're looking at any and all avenues to move this into the patients and get this to the clinic as quickly as possible.
And Jenny, I'll jump in there
Yeah.
real quick. The beginning of the year, we had hoped to talk to you guys more about a partnership that we had been working on for the past year. We did end up signing a deal with a small startup for this program around February, and we're about to announce that to you guys. Unfortunately, geopolitical headwinds, changes at the FDA meant their investor base did not pull through with the funding that they had hoped on the timelines that they had originally hoped. And so we've seen that it's not specific to this program, not specific even to that startup. Unfortunately, this has been a headwind that most investor kind of that landscape has been tentative of this year, just given what's going on and all the changes that we're seeing. So we're still trying to find a path forward with them, but it's forced us to consider, we have much of the funding in place. I wouldn't say 100%, but much of the funding in place to get through our non-clinical portion of the program. This is everything right before clinical trials. We're now just trying to piece together how we identify the funding for clinical trials. And so we've been working on that this year with that partner. We've been looking at alternative paths. It's something we're actively exploring. I don't have more to update on that front right now, but we are encouraged by some of the discussions that other groups in the rare disease space have had with the FDA about an openness to alternative paths. So when we talk about current timelines and our clinical trial plans, those were clinical trial plans that were put in place in discussion with the FDA during our pre-IND meeting in January. Those still stand today, but we have to identify the clinical trial funding in order to move those plans forward. And in parallel, looking at alternatives. So some of the things we're going to talk about, especially in the question section, may end up shifting if we do find alternative paths and are not able to identify traditional levels of funding. So I know that's not concrete information on what the path is going to look like, but during the next year, we'll hopefully finish all of the non-clinical studies and then sort out what the most likely path will be to clinical trials. But this space has gotten pretty creative of late, and we are trying to take advantage of any and all creativity as far as funding and partnerships to ensure this keeps moving forward.
Okay. So this is the current timeline and where we are to date. All of the green activities have been completed. The dotted vertical line is where we are at today, and everything in the orange or salmon color are activities that need to be completed, preclinical activities that need to be completed. The green check marks we have funding for, and so those opportunities are secured and we are moving forward with those. The gray check marks mean that those activities are partially funded, and then if you don't see a check mark next to a orange arrow, that means we do not have funding for that activity. So we are very, very close. We will be initiating, again, that safety study on Friday, dosing in early 26, and would plan to have a full report and information on that safety study by mid-year. In the meantime, we will also be moving forward with histopathology on that study, as well as biodistribution and transgene expression to understand the vector. And then a couple other assays that we do need to find funding for are highlighted in orange. So if we had all the funding available today, we would be looking at an IND submission in 27 and getting to clinical trial. So that's where the current timeline is. We are very, very close. This is really the final stretch on the preclinical activities. So you guys have done an amazing job to help us get to where we are.
Yeah, Jenny, I want to just, for those who are not as familiar with what these studies represent, just take a second to kind of summarize where we've been with the program. So we've generated data that shows that the treatment, the AAV, and this, if you see OT-004, that is what we call our drug name for this program. That's the gene therapy that would be administered. We've shown that's effective in a mouse model with vision loss caused by RPGRIP1. We've been through manufacturing with our manufacturing partner is Andelin, and been able to manufacture really high titer, good quality product. So we have a developed manufacturing process now. We've generated the vector that's going to be used in our toxicity, what we're also calling our safety studies, which is this next step in that right across that vertical line. So those are big. The manufacturing piece and the toxicity studies are very big, expensive pieces in drug development. They're also really the last stage pieces. And so once we have this safety study completed, early mid next year, it's really just about analyzing the data and setting up what we call the analytics. So it's a lot of what those multiple orange arrows are, is how do we look at the safety? We look at the eye during the safety studies. We mirror how we do the administration, how we will eventually do it in clinical trials. All of that's mirrored to look at how safe the profile is at different doses. And then look at the distribution where the vector goes in the eye. And it will distribute out into the body to some degree, but the eye is largely isolated when we do that administration. But it's looking at everything downstream, really just to ensure that we have as safe a dose as we can going into clinical trials. But It's a lot of arrows, but this is kind of the home stretch now. So we're excited to be in this stage and really appreciate your support over the past 5 or 6 years.
And I will pass it off to Heather.
Thanks. Do you want to switch to the next slide? Oh, thanks. So, Ashley and Jenny have mentioned this, but the reason that we are here at this great place is because of the support that we've gotten from y'all on the community, from the community, and the contributions that you have sent in, but also the relationships that you have helped us make. In the last year, especially, we've had some support from new foundations, and we've had a couple matching gifts that have made a significant difference. So, a company or a foundation has matched a gift made to the program, which has been tremendous. And so, that is fantastic. And you can see the thermometer on the right. For those of you who are here in March, this is looks familiar, but the numbers are getting a lot better as far as what we are still looking to raise. But to get to IND, the we've been had great success in leveraging our nonprofit status to help reduce some of the costs. So we have that's less money that we have to raise, which is also fantastic. So to get to IND, we're estimating it's 1.17 million, but that number, Jenny is constantly trying to get us leverage that nonprofit status to help that number come down. But that's what we're looking to raise to finish up some of those orange arrows that were on that last slide. I did want to give a shout out to a couple of folks who have done a tremendous job with the fundraising pages. We have a partner every.org, which is a website that allows folks to set up fundraising pages. And we've had a couple families that have set those up. They tell a little bit of a story about the person in their family that has RPGRIP1, and then they send that to friends and family, and folks make donations, and that has been very successful this year. And so if you are in a place that you can set one of those pages up and let others know about what we're doing, we encourage you to do that because we've seen a lot of success this year. And and along those lines, if you work for a company that does matching gifts, we've registered, Odelia has taken time this year to register on several of the matching gift platforms. So if you make a donation and then your company matches a gift, they'll probably have a online portal. You log in and you search for us, and then the company will make a matching gift to us. And so we should be in most of those databases now. Which would make it a lot easier to make matching gifts. And that doubles the gift. And those go a long way to help us also with the funding of these the different steps we need to complete to get to IND. A lot of these ideas on the bottom. So we're obviously still looking to raise more money. And we have on our website under the support section, there are a few PDFs with instructions as well as links to other places to help raise money. There's obviously a link to every.org and to the donation page. And on the donation page, people can designate the funding as for the RPGRIP1 program. And then it automatically goes to the gene therapy program. There's a organization that we've worked with in the last year. It's called Group Raise, and they have partnerships with restaurants all over. Every restaurant I feel like that anyone goes to, including things like Krispy Kreme, Honey Baked Ham, and you can set up a fundraiser with we had a family do a fundraiser in Texas, and you just tell your friends and neighbors to go to the restaurant on a particular day, and then the restaurant donates a portion of the sales that night to Odelia. And it's all of these things that are adding up to make this big difference in this thermometer that you're seeing. So I encourage you to do that if you're able. If there's a restaurant in your town that you can set up, it's just a simple process to go online and sign up and pick a day. And the other thing we've been trying and having making a little headway on is some foundations and we're trying to expand our awareness into angel philanthropy. So people who want to um, give money to kind of unique programs as well as family offices. And those are offices that help families with high net worth with their investments, but also with their donations. And so some of those families are interested in gene therapy and medical research, but they don't necessarily know where to give that money. And so we're trying to raise awareness about Odelia with those folks. So I say all that because if you know someone in a foundation or a family office or anywhere that might be interested in Odelia, we can send them a link to our impact report from the website, and we just can get awareness out about what we're doing, and then hopefully that will generate some donations to the program. So more of this is available on the website. Y'all have my address, my email address, and you can certainly reach out and I can answer any questions. That's probably the easiest way.
Jenny, do you want to go to the we skipped over interest because we were trying to get that link out. There's I think we have a PowerPoint slide with email addresses for everybody on the phone call.
I think it's at the
Odelia's.
end.
It is at the end. I can flip over to it real quick.
Yeah, so if you have any questions, just so you make sure you have our email addresses, feel free to reach out. You may you'd be used to seeing Catherine post Demont from our team. She is actually on parental leave now, but she'll be back early next year, so she couldn't join us today. But feel free to reach out with any questions. And then, do you want to jump to the question and answer section?
Yes. All right. So the
All
first
right.
question, I can kick it off, is the timing of the next phases, including human trials. So we are in the final stage of the preclinical phase right now. So we would consider the toxicology slash safety study to be this final piece, IND enabling study for preclinical. Once this is complete and we have the data, um, we're good to go on the preclinical side. So we quickly move to clinical phase activities. Ashley, do you have anything to add?
No, I think that makes sense. I think much of what Jenny's trying to align right now is how quickly we can get to clinical trials, and we're trying to, on the other side, identify the funding to do so, or alternative paths to clinical trials. So, in that case, some of the questions, we got a lot of questions around clinical trials and has a location been determined? We haven't pre-selected a trial site yet. Most likely we are looking at a single trial site, and that's pretty common with gene therapy because it's an invasive procedure in a rare disease population. For clinical trials and consistency of that delivery in an early phase 1/2 trial, we are likely to look at one site, and that may be dependent upon funding and timing where that would be. We've primarily to date interacted with the FDA in the US. We are looking at other regulatory regions to understand if there's other ways that we can kind of navigate the financials. There are some incentives of running trials in other regions. There's just different expectations sometimes as well. And so we're looking at all of those paths, we're looking at different partnerships. So those would likely play into that decision at the end of the day. And what is the age or limit requirement? So again, we have a clinical trial design that was well received by the FDA during our pre-IND meeting earlier this year. If we explore alternative paths or different regulatory environments, this may change, but I'll speak to at least what the plans are as they sit today. Our current inclusion-exclusion criteria for the first clinical trial for RPGRIP1 would be in a age range from 10 to 17 years old. And that's with a confirmed diagnosis, vision loss caused by RPGRIP1, So having that genetic report, I think there's a question about how best to prepare for future clinical trials. Making sure you have that genetic report, you have a copy, you've spoken with your genetic counselor and your physician. There's an understanding that confirmation that everybody here is here because of an RPGRIP1 mutation. Things like that are just important for record keeping because that will be an inclusion point. There's an aspect of the inclusion criteria around confirming that there are cells to treat in the eye. So we have to be able to deliver the gene therapy to photoreceptors that are intact. And this is part of the selection process around the age group of 10 to 17-year-olds is we do think that there's a high retention of photoreceptors and not a lot of actual photoreceptor death or loss at this stage. So from what we have understood from the clinicians we've spoken to in this space, I would say there's thought that in this age range, you don't see a lot of loss yet. So we are trying to treat initially in a patient population where there's starting to be functional vision loss, but not loss of the photoreceptor. So, and this goes to some of the other questions we received. We think the potential here is first and foremost to stabilize function at the point of delivery or receiving this treatment. It's a one-time treatment. And so the initial goal is to stabilize function at that point. But we do think that the patient population where if we go in early enough, if there has been vision loss, we think it's largely due to a functional loss, but not the death of a photoreceptor. So we think there is the potential for some regain of function. The clinical advisors we work with have seen this in similar programs, and they think, given the biology here, that is also a possibility. But those are very, very, very hard questions to really answer until you get into clinical trials. So we're hopeful that there's also a regain, but at a minimum, a stabilization of function at the point of delivery. All right. So I've answered a few of these. Let's see. Is it true that a person can participate in one trial or treatment only? If someone participates, will they be unable to participate in other clinical trials? It depends on the clinical trial. Usually it can be hard to participate in an AAV trial and then in other programs, but it really depends on if there's we're very much aware of like the NAC attack trial, using NAC in the eye in the context of retinitis pigmentosa. We know a few patients from other programs that participate in that trial. That's a small molecule that wouldn't necessarily preclude them from participating in the context of maybe a gene therapy later on. So it really depends on the biology. It depends on the nature of the endpoints and the readouts. But once we step into running a clinical trial, all of this will be reviewed with the community in more detail. So we'll be able to provide educational materials. This is part of the consent process. We're happy to answer questions. How can families prepare for a future preclinical or clinical trial? So one thing I mentioned was having access to your genetic report, having a copy of that, making sure you've reviewed it with your physician and there's an understanding and an agreement that your vision loss is caused by mutations in RPGRIP1 and that there's not ambiguity or question marks around that. Heather, Jenny, we were talking about this question a little bit yesterday and whether there are other things. I think one thing that comes to mind is making sure you follow us on social media and our newsletter. You're receiving updates for Q&As like this because this is where we would start to communicate clinical trial plans going forward. And educational materials will be a big component. Answering your questions about gene therapy and the enrollment process and consent. Anything else, Jenny, Heather?
No, I think you touched on the big pieces, but making sure you have, your medical records and genetic reports ready to go and having those available. I know some hospitals and medical facilities take a while to get that stuff, and you have to go through different offices.
Yeah. I think it, having a touch point. I think we've heard of variable. When we've spoken to our patient advisory board about this program, I think we've heard a very variable response to how often patients were going to see the ophthalmologist to get checkups. And in many cases, I think earlier on when kids or patients are younger, that may be a yearly process, but we've heard from older patients that they start to drop those visits. So I think just re-engaging with whomever that focal point is for you to start asking these questions about eligibility and whether this would be a good consideration in your case, and reconnecting with that doctor to see if there's any reevaluations that need to be done. I think that's always an important consideration as well. And starting that conversation now and in the next year would be good timing. All right, so funding and timing of subsequent phases to availability to general population. So we're hoping in the current plans to very quickly pivot if we have promising results, if things look safe. What we'll work through initial in the current clinical trial design is establishing what we call maximally tolerated dose. So that's trying to figure out the dose that safely, effectively treats patients, showing the safest profile, less adverse events. And happy to answer if anyone has specific questions on, what these words mean or how this actually plays out in clinical trials. We are always happy to get on the phone and answer questions. But we're trying to work towards in our phase 1/2 declaring a maximally tolerated dose, which would be what we take forward into a broader trial or into younger patients outside of that 10 to 17-year-old range or older patients. So we start with 10 to 17. We work through the dose, identify the best, most effective, safe dose, and then the hope is to expand to younger populations and older populations. And as a rare disease company, we're looking to do that as aggressively as possible. Usually, traditional pharma moves through a phase 1/2, phase 3 into maybe approvals. And we're looking at and are aware of other programs that have moved very quickly off of a phase 1/2. If the data looks great, if we're seeing actual vision loss stabilization or recovery in a clear way, we would seek to be aggressive about broadening that to a general population as quickly as possible. But it's going to be depending on the data and how robust that plays out.
I can jump in with question
6.
6, yeah. Estimated timeline for completion of the preclinical safety study. So there's also a question in the chat about that. The safety study is not a human safety study. The one we are currently talking about, the one that's about to begin, that is the final preclinical safety study. So not in humans quite yet. And that should be completed by mid-year next year. We also have, I'm going to throw this question out there. It's from Aaron Ashley. It's a technical question, but I'm trying to pull it up.
I'm looking at it. Actually, I can read
Okay.
it. So, how DNA bases get replaced in the RPGRIP1 gene. So,
Yeah.
we do call this an AAV gene replacement therapy, but we're not replacing the mutation directly in the genome. That would be more akin to editing, um, gene editing techniques. We are delivering a whole new copy of the RPGRIP1 gene to the eye. So the patient will still have the RPGRIP1 with the mutation in it, which we don't consider to be functional. And what we're putting in is a full length wild type, so without that mutation version of the DNA. into the cells that that's delivered to. And so the thought is that new gene will produce a transcript that's then translated into our functional RPGRIP1 protein, and we'll be able to rescue the function that's been lost from the mutated RPGRIP1.
Okay. For question 7, we've touched on this a little bit, which countries are expected to be prioritized. I think right now that's still up in the air. As Ashley mentioned, we've primarily interacted with the FDA up until this point, but we are looking at any and all paths to get this to clinical trial. So we're not ruling out other parts of the world, and we're looking at ways to get this to the patient. And for question 8, Ashley touched on this a little bit, but what criteria or process for patients to be considered for participation? Just want to go through the inclusion criteria as it stands right now, right? This was the synopsis we put together for our pre-IND meeting with the FDA. So there's still an opportunity for it to change as things develop with our clinical plans and funding situation. But where it stands right now, Ashley.
I'd have to bring up our latest. Maybe we can move forward while I'm bringing up our what
Yeah,
we discussed during the pre-IND. If you want to go to the next slide, I'll pull that up.
sure. And then for question 9, how fixed is the current lower age limit of 10, and how soon could younger children be included once more data becomes available? So our current clinical trial design for the phase 1/2 has an inclusion criteria of 10 to 17. So we would start with that age group, and then once we see that that is safe, we would be able to expand that. With the current clinical trial, we're looking at a couple of years, but we, because the clinical trial design is still in flux, I think there's a chance that may change just depending on the funding situation and what alternative clinical paths we might have to pursue to get this to patients sooner if the traditional funding schemes don't work in our favor with this current economic climate.
Yeah, and I can speak to real quick I have our inclusion exclusion criteria up as it currently stands. So this is again a synopsis of the clinical trial plans. We discussed it with the FDA. They were receptive to it. This is not the final clinical trial protocol that would end up being used. So these protocols and the establishment of what we'd end up using do get pressure tested quite a few times. And so that this could change. But I think we spoke to most of these earlier. Age range of 10 to 17-year-old, confirmation of RPGRIP1 mutations, confirmation of photoreceptors in the area where we'd be looking to deliver at an adequate level to potentially benefit. And I think this is the question mark when we move into not the younger population, because we think there'll be even more retention of photoreceptors, but as you age in an older population, we do think there's still potential for benefit, but the progression becomes more variable. And so when we're trying to see how effective this therapy is, we want to go into a population that we know has the most potential to benefit, and then expand into an adult population, an older adult population, where some of those variables have been worked out, and we'll be able to better understand how to deliver this. Because I think it just becomes, in a rare disease population, a matter of really identifying patients and data and clinical understanding. And then we have a requirement, that's our ellipsoid zone is really linked to this treatable photoreceptor area. And then I'm skimming our exclusion criteria, all linked basically the same thing. There's some exclusions we have currently for pre-existing conditions that would make someone a bit more at risk for gene therapy delivery, like immunodeficiency, prior gene therapy administration, so that we have that as an exclusion if you've had a previous gene therapy, which I don't think would be the case in this population, but if that is, please let us know. history of allergy or sensitivities to medications that we might need to use during the administration, which is a surgical intervention, or other CNS diseases that would put someone at risk for receiving anesthesia or the gene therapy or any other medications around it. So again,
And
feel free.
as
Go ahead, Jenny.
no, I was just saying, as we get closer to this point, like this will be more formalized and we'll put together, all the inclusion, exclusion criteria for this population. So there's no ambiguity about what's to come and what next steps are. But just to answer some of these questions now and highlight them, we wanted to address it.
Yeah.
And then we can touch on question 10 again. I think you answered this earlier, Ashley, but is this gene therapy expected to stop further degeneration, or other indications that it could also restore vision?
Definitely bottom line stop degeneration at the point of administration, but I think we have high hopes, both indicated from the biology, our clinical community, and what clinicians have seen in other similar settings for subretinal AAV delivery, is we're hopeful that there will also be restoration of some vision. And then insight on cost per patient. We don't have any insight at this point. It's very hard to predict this. So Luxturna, I think, is about four to 480,000 per eye. But we're not aiming to be anywhere near Luxturna. That being said, if we do partner the program, it's going to be largely up to that partner and what their pricing looks like. But there are different pressures that change over time and influence that cost. Our hope is that this stays more affordable. But our primary goal at this stage is really just to ensure that this gets to patients as quickly as possible. So whether that's through partnerships, other funding avenues that may have an influence on cost down the road, but hard to say at this point, really.
And then there's a question in the chat. Is there a possibility of including older generations with photoreceptors?
Yeah, our ultimate goal is to go into the full spectrum of RPGRIP1 patients. And so the treatable, because the AAV is treating and delivering RPGRIP1 into photoreceptors, really the end goal is to deliver it to all patients who have a retention of photoreceptors. So that's the aim and our hope. We don't know the timeline ultimately for how quickly that will play out, but our hope is to expand once we get the initial data that looks good
Ja,
in the 10 to 17 year range, we'd quickly
ja,
expand
het
in
gaat met
both
name
directions.
over wanneer.
Okay.
Yeah.
So I know it's frustrating to not understand those timelines. Uh, but I do think as long as there are retained photoreceptors, we do not see a reason that those wouldn't be treatable ultimately. Any other
Okay.
questions? And feel free, you can either put it in the chat or feel free to raise your hand if you want to ask us directly. You don't have to come on video either.
No. Okay.
Jenny, would you mind going to the slide with our emails just one more time in case anyone wants to reach out?
Sure.
We also have the info email, which we monitor, so feel free to reach out to anyone. We have these 2 to 3 times a year, depending on updates. Um, we're we're hoping, I think next year, just given the uncertainty around funding and this program reaching clinical stage, we're hoping to have probably an update on what that might look like in gonna get ahead of my skis a little bit here, but probably in the second or third quarter, probably around mid. I'm hoping before middle of the year, but I don't want to, you know, put together a Q&A without having something to actually update y'all on. But we will have the safety data coming back. When's the timeline for that, Jenny?
Yeah, that'll be second quarter. So
Okay.
we'll have safety data to update on and hopefully a clear path to clinical trial and
But
clinical phase.
if anyone has questions in the meantime, please feel free to reach out. Um, we are, as I said in the beginning, we've kind of reiterated actively exploring alternative routes at this point. So if anyone has contacts in the life sciences or investor space or philanthropy that we haven't spoken to, please feel free to make that introduction. And we're finding the rare disease space to be more and more creative. We're open to very, very creative partnerships. So I know we'll get there. We're not sure if exactly what we had planned with the FDA earlier this year is exactly how it will play out, but we are pursuing any and all paths to bring this to clinical trials as quickly as possible. So we appreciate your support and let us know if you have any questions about the program. Um,
And there's
Ash-
one final question that came through the chat.
how do we know that the child only has light receptors and that the treatment is unsuitable? So I think that the question of suitability is really something you want to talk to your the physician that's following your child's case or your case, depending on your age. Whether it's your ophthalmologist, usually there's a number of doctors you end up seeing in this process, but whomever is the one delivering, if it's a Leber congenital amaurosis diagnosis, we know some of our community has juvenile retinitis pigmentosa diagnosis, some have a cord diagnosis. So there are multiple diagnoses we've seen with RPGRIP1. Um, whomever you who's delivered that diagnosis and knows the link to your RPGRIP1 mutation and ideally follows your case or your child's case over, annually, or maybe it's every few years you connect. We've heard different timelines from different families. Whoever that individual is, that's who I would start this conversation with. Happy we've been connected with quite a few doctors over the years that our community has seen. Um, if there's any questions we can answer on their part, feel free to put them in touch with us, and we can help, kind of guide our current inclusion exclusion criteria and what that may look like and how it relates to their case. But that doctor would be the best person to advise on that at this stage. Any other comments, Jenny, Heather?
No, not from my end.
Okay.
We just want to say thank y'all for coming. We apologize for the little snafu with the link, but we're glad you are here. And if you know folks who want to watch this, we will post the recording on YouTube and we'll send out a link to that as well in the next day or so.
Yeah, we can usually get it up pretty quickly afterwards. So look for that. And we have all the prior Q&As as well up on YouTube. So actually, we should add that to the link on this slide. That would be helpful. But yeah, please follow us. There's a way that it will update you anytime we post new material on YouTube as well. Okay,
All right.
thank you.
Thank you
Appreciate
all.
all the support and y'all showing up today. Again, let us know if you have questions and we'll be in touch. Thank you.